So the next talk is Dr. IL40 Banhopadhyay, and she is in the Department of Zoology. She is going to talk on the effect of small molecules on protein folding manuscript. Rania, please.
Certain small molecules that can act as chemical chaperones on protein folding landscapes. So, proteins are the most diverse and complex biological molecules, and inside the cell, proteins are synthesized from ribosomes as linear chains. However, these linear chains can be as long as 1,000 or more amino acids, and therefore, folding of these proteins inside the cell is a fundamental question that still needs to be answered. Not just that, these proteins must be kept in a proper three-dimensional conformation to be biologically active, and the cell expends a lot of energy to make molecular chaperones and other helping proteins that would help certain proteins to fold. And in case these proteins are not able to fold, in which case they would aggregate, and these aggregated proteins can lead to certain diseases, for example, neurodegenerative diseases like Alzheimer's and Parkinson's, where proteins like tau and alpha-synuclein, they aggregate. And apart from that, even in metabolic disease like type 2 diabetes mellitus, it has been seen that there's a protein amylin which gets aggregated and causes beta pancreatic cell death. Therefore, the cell has to expend a lot of energy to keep the protein homeostasis, maintain the protein homeostasis, because this is crucial for the cell to survive. Now, as I talked about these linear polypeptide chains, they have to fold into the proper biologically active three-dimensional conformation. These linear chains undergo folding and they travel through an energy space. This energy space can be shown in the form of a funnel, where the horizontal axis is basically the entropy of the molecule, the flexibility of these linear chains, and the vertical axis is the enthalpy or the energy, which slowly comes down as the protein explores all the conformational spaces and forms native contacts and comes down in energy and forms the most stable native conformation. However, as on the left-hand side panel, you can see the funnels are not very smooth. For most of the proteins, especially the proteins that are longer than 100 amino acids, this folding is by itself, the folding is difficult, and therefore the terrain of this funnel is quite rugged. They have to make their way through this rugged terrain in order to reach the final native conformation. On the right-hand side panel, you can also see that proteins that tend to misfold, they generally aggregate because they are not able to slide down through this funnel to the native conformation, and on the contrary, they would go to some local minima, energy minima, where they would make some non-native contacts, and because of that, they would aggregate either into amorphous aggregates or into amyloid aggregates. So therefore, for the cell, it is imperative to make sure that the second law of thermodynamics, that is the free energy of the folding of the molecule, must be ensured, and this is where two components for this free energy comes into picture. One is the enthalpy of the folding, and the second is the entropy of the folding. Alright, so from here, we know that inside the cell we have a lot of proteins called molecular chaperones like heat shock proteins, an entire repertoire of these molecules that would help these any newly synthesized protein to fold. Apart from that, we have also found out that there is literature that mentions that along with these molecular chaperones, there is a whole repertoire of chemical chaperones that might also help proteins to fold. In vitro, these have been studied, and in the category of chemical chaperones are osmolytes. These are small organic molecules present in the cytoplasm that basically help the protein maintain its osmolarity. Apart from that, so the category of these molecules are sugars, amino acids, and methylamines like TMAO. So there are studies that show that these molecules stabilize protein and therefore must be helping the proteins to fold. What we asked is that what is the mechanism by which these chemical chaperones help protein fold. Also, we asked whether all the chaperones that have been identified, whether they do this by the same mechanism or whether different chemical chaperones will have different mechanisms by which they help proteins to fold. So as I mentioned in the protein folding funnel, we have two components for this folding. One is entropy and the other one is enthalpy. So if there is a protein that has difficulty to fold, either it has got trapped based on its enthalpy or it has got trapped because of an entropic barrier. And this is what we wanted to ask for different chemical chaperones, taking a few test proteins, that whether the folding barrier, whether it's entropic or enthalpic, which kind of small molecule is going to help that. Now why I am talking about this here is because in case we understand how these mechanisms are, then we can probably design de novo molecules that can help in preventing protein aggregation and stop those diseases, or at least help these proteins to form correct conformation and not go into aggregates, because of which we have these kind of protein misfolding diseases. So the two test proteins that we have used, first and foremost we have used motors binding protein. There's double mutant of that, which is established to be a slow folding protein and has a kinetic barrier from the unfolding to the folding properly folded stage. And to corroborate our results, we also checked the same small molecules on the GFP protein. The candidate chemical chaperones that we have used are trimethyl epinoxide, which is a proven chemical chaperone, and along with that we have used trehalose, proline, and C. So the first experiment, the first question we asked is how can we figure out if the double mutant motors binding protein, what kind of a kinetic barrier it has, and what kind of assistance it is getting from TMAO. So here on the left hand side we have this crystal structure of MEP, where it shows the two residues at position 18 and 296 that we mutated into cysteines, so that we can have, and we know in the crystal structure that these two residues are closer to each other in the folded form. So we put cysteines there and we carried out quantitative cysteine reactivity. What we did basically in this reaction is that we unfolded the protein, reduced it, and then we allowed it to fold in oxidative conditions in the presence or absence of TMAO. After certain time intervals, we quenched the reaction using biotin-malamide, because of which it will, the biotin-malamide will go and bind to the unreacted cysteines and will quench the reaction of disulfide formation at that point. And then through immunoblotting, we were able to see the effect of TMAO. So compared to spontaneous folding, when we looked at the effect of TMAO, we found that the disulfides between 18 and 296 were forming much faster in the presence of TMAO as compared to in spontaneous folding, telling us that in the presence, in the environment of TMAO, the protein backbone is coming closer, and therefore the disulfide bonds are forming faster. So this gave us preliminary evidence that TMAO is helping the protein fold by reducing the entropic barrier, because it is reducing the flexibility of the peptide chain, as a result of which it is leading to a faster conformation attainment of the protein. To deconvolute it further, we carried out Arrhenius analysis, where we were able to differentiate between the enthalpic and the entropic barrier. So in the Arrhenius analysis, we took two different concentrations of TMAO and compared that with that of spontaneous folding across 7 different temperatures, and compared that or plotted that against the refolding rates. And by fitting this data into the Arrhenius equation on the right hand side panel, you can see the rho value, which I have highlighted in yellow probably. There we can see that the rho values are decreasing. So as the rho values are decreasing because of the presence of TMAO, it shows that the entropy is being controlled by TMAO. And similarly, we have also shown that the disulfide mutant, where the two components 18 and 296 are coming closer by itself and causing decrease in entropy, that is being affected less by TMAO. So this shows this gave further evidence that TMAO is basically working on the entropic barrier and decreasing that. So based on these evidences, we carried out a high throughput screening to investigate if there are, apart from TMAO, there are other molecules, small molecules, that would do the same or similar kind of function for the DMMAP protein. On the left hand side, we have the heat map for different small molecules. So we have taken multiple amino acids, some sugars, and tried to see the refolding rate as well as the refolding amplitude to see what kind of effect these molecules certain parameters, we selected, apart from TMO, we selected serine, proline, and trehalose for our further studies. On the right-hand side panel, you can see these are the refolding traces of DMMBP in the presence of different amino acids on the top right-hand side corner, and at the bottom we have the effect of different sugars on DMMBP folding. So from here, from the heat map, from the data that we had got, we selected 2 amino acids, proline and serine, and we selected trehalose as well as TMO that we were originally working on, and carried out the same Arrhenius analysis to see what kind of influence they have and whether we can deconvolute the enthalpic and the entropic barrier between these two, effect between these osmolytes. So on the left-hand side, we have the Arrhenius analysis as had been done previously for TMO, and on the right-hand side again we have the highlighted column where we have the row values, which tells us that trehalose has similar function as we had for TMO, whereas proline and serine do not work in that way. On the other hand, proline and serine are the way they are bringing faster folding is by working on the enthalpic barrier. So we clearly see that different chemical chaperones have different modes of carrying out their action of helping proteins to fold. Prior to this, there were no evidence which would specifically tell, comment upon what kind of effect a particular protein, a particular amino acid or osmolyte is bringing in order to fold the protein faster. Alright, so clearly we could figure this out, that from the threshold value on these two boxes that you can see, serine and proline are lower because they are showing assistance by changing or reducing the enthalpic barrier, whereas TMO and trehalose, they are helping the protein to fold by reducing the entropic barrier. To corroborate this further, we carried out single molecule FRET experiment to see what kind of populations are generated in the first 100 seconds of refolding of DMMBP. In order to do this, we selected 3 pairs of, we made 3 pairs of cysteine mutants. Those are mentioned on the left-hand side panel on the crystal structure of DMBP, and we tried to see that as the proteins fold, what kind of populations are generated in the first 100 seconds of the protein folding in the presence of different molecular chaperones versus spontaneous. And on the right-hand side panel, we have the FRET efficiency distributions, which tell us that if we look at the parameters for all three pairs of mutations, we see that the distribution that we have of populations in each of them are different from what we have in spontaneous. At the same time, if we compare them, which I have in the next slide, if we compare the effect of proline and try to superimpose that on TMO, trehalose or serine, we find that for TMO and trehalose it is different from that of proline, whereas in case of serine it is similar to what we have for proline. And again, this was like further evidence at the single molecule level that the mode of action of TMO and trehalose is similar, and whereas the mode of action of serine and proline were different from that of TMO and trehalose. Now, at this point we wanted to know that this kind of categorization that we have between these chemical chaperones, does this hold true for any other test protein that has a folding issue and can be fixed by the same mechanism, or whether it varies from protein to protein? And therefore, we used the enhanced green fluorescent protein, and we carried out Arrhenius analysis for all these for this protein using 24 different chemical chaperones. And we found out, to our surprise, that the mode of mechanism now in case of GFP was different from what we had found in case of DMMBP, which more or less told us that depending on what kind of protein we have and what kind of folding intermediate or folding kinetic trap it is into, the effect of the chemical chaperone would be different. So the kind of entropic barrier removal that we could see for trehalose and TMO was not present in case of GFP, probably because the structure of GFP is different, and the kind of assistance GFP required for a faster folding was the requirement was different as compared to that of DMMBP. So based on this, we could conclude that chemical chaperones, they function in a much diverse way compared to what was thought earlier, and therefore much more studies are required to be able to categorize this. The conclusion we could draw from our study was that the mode of assistance that these chemical chaperones could provide depend on two factors. One, the kind of refolding barrier that, or a folding barrier that a protein would have, and also the nature or molecular properties of these chemical chaperones that are being used. So therefore, any chemical chaperone would can assist a protein to fold either by removing the enthalpic barrier or removing the entropic barrier, and that would depend not just on the on the chemical chaperone but also on the type of folding intermediate or trap that the protein is into. Based on this data, we can project from here that we should be able to design de novo chemical chaperones that should be able to have a synergistic effect together. One component taking care of the entropic barrier, the other component taking care of the enthalpic barrier, and together they might be able to ameliorate the kind of aggregations that we see in different kinds of proteins, be it amyloid proteins, amyloid aggregates, or amorphous aggregates. And future directions would be to try them out in vivo systems. So I would like to acknowledge my postdoctoral mentor, because this was my postdoctoral work, Dr. Kaushik Chakraborty in IJIB, CSIR IJIB, my lab members and funding agencies, DBT, India Alliance, and CSIR. Thank you.
Very good presentations. These are we working away from native conformation, all these proteins?
Yes, of course. So this is the pathway from where the protein starts to fold. So once it is translated.
I know that whatever the way you are analyzing the folding.
Yeah. So what we are doing.
I feel that it is away from the native thing. What is happening?
I would beg to differ, because what we are doing is we purify the protein, then in vitro we are unfolding the protein, and then we are allowing it to fold. So this is in a buffer system, and it is folding now in the presence of certain other molecules that we are adding to the buffer system.
Those are not a native conformation, right?
So the protein is trying to attain the conformation.
Do we have the CD spectrum for to ensure that whatever the protein under better not to touch, right? Naturally, protein mutation under it makes a serious conformation changes.
Proline, I did not do any proline mutation. Proline was added into the environment to see if proline would have an effect on the folding of the protein.
This folding,
these
refolding,
yeah.
Do we have the CD spectrum for it?
We have the CD spectrum for the fully folded protein, we have a CD spectrum for the unfolded protein. No, I do not have the data here.
But for GFP, you are getting the green fluorescence, isn't it? So that's
yes,
the activity.
Yes, sir. So the
correct folding, or do you require a chaperone there?
No, sir. So we have put cysteine there to see if disulfides are forming faster, because the environment has changed.
So faster is fine, but suppose there are multiple cysteines, how it is going to help that the correct cysteine is that one and not this one?
So that does happen, and for that we have cysteine protein disulfide isomerases.
That's what I'm saying. So you need the chaperone.
Yes, sir. You will need that enzyme to carry out that. But in case of DM-MMP-FS protein we have just selectively put cysteines there. So the cysteine disulfide that is forming, it is that particular 8 and 296 that is forming the disulfide bond, not any.
All right.
Just
last question, quick.
Okay, I'm a little curious. You mentioned trehalose. So does it have anything to do with the fact that trehalose is a non-reducing sugar and it serves as an antifreeze?
There are multiple sugars that have been shown to have this effect on protein folding. So in many cases trehalose has been shown to do this. I cannot specifically comment on the fact that it is a non-reducing sugar and therefore it would be having this effect. But presence of these polyols have been shown to have better folding effect on multiple proteins. It has been shown for amyloids also.
Just adding on to that, many of the tardigrades and some of the organisms which can withstand extremes, they have this, as you rightly said, antifreeze and all. Yes.
Marine animals and animals, they are in deep sea. they have to survive under pressure. They generally have a higher concentration of TMU, beta-in, taurine to take care of, the correct conformation of the proteins are being, maintained inside the cell under high pressure and higher salinity. Yes, cryoprotect and piezo.
Thank you, Dr. Nandini.
Dr.
Sorry.
He makes my photograph digital photographs.
Okay. Wait.
Thank you.
Please.
I'm not ACSR, but